Hi everyone, today we're going to take a look at the Calvin cycle, which is the second stage of photosynthesis, and we're going to look into up until the point where we produce a G3P molecule. As we've spoken before in our previous lecture, photosynthesis divides itself into two stages. First, we're going to have the light-dependent stage, and second, we're going to have the light-independent stage. Calvin cycle is pretty much the light-independent stage of photosynthesis, and Calvin cycle, what it does is it essentially uses the energy from ATP and the reduced hydrogen carrier NADPH that were produced from some of our previous light-dependent reactions, and it's going to use that energy. And what it essentially does is it's converting CO2 into glucose. That's the ultimate goal of the Calvin cycle. And in this video, we will be exploring the Calvin cycle up until the point where we make G3P. Now, the Calvin cycle, where does it occur primarily? Well, the Calvin cycle pretty much occurs in the stroma. More specifically, it occurs wherever RuBP is located, because RuBP is kind of like the primary ingredient of what happens in the Calvin cycle. So we can say that the Calvin cycle occurs in the stroma, wherever RuBP is located, and its primary function is to convert CO2 into glucose by using energy that's produced from ATP and NADPH, which are molecules that we have created from our previous light-dependent reaction of photosynthesis. So let's get right into it. Okay, so what's going to happen first is we are going to start with this stage. Now, what happens in this stage is carbon dioxide is essentially fixed into a carbon-5 molecule, and this carbon-5 molecule is called RuBP. And RuBP pretty much stands for ribulose bisphosphate. So we're going to have this 5-carbon molecule, RuBP, and to it we're going to kind of fix carbon dioxide. And this process of fixation is called carboxylation. And because we fixed a 1-carbon molecule, the CO2 molecule, with RuBP, which is a 5-carbon molecule, what we're going to have is an unstable 6C, a 6-carbon intermediate molecule. Now, intermediate meaning that it's kind of in between stages. And the reason why this molecule is so unstable is because it has too much energy. And we also need to really clarify that the term excited is used to represent the state of electrons having too much energy, but for other molecules, we just use the term unstable when they have too much energy. Now, because this molecule is so unstable, it's going to be immediately broken down into two different molecules of glycerate 3-phosphate, which we kind of abbreviate into G3P. Now, G3P is a 3-carbon molecule. Now, this process of carboxylation up until creating the unstable 6C intermediate molecule, and then breaking that molecule down into the G3P molecule, this step of the Calvin cycle is pretty much catalyzed by an enzyme that's called Rubisco. More specifically, it's RuBP carboxylase, which is, as you can see by the ending ase, it's an enzyme. Now, Rubisco is just one example of the many enzymes that are involved in the process of Calvin cycle, but in our IB curriculum, it's a must-know, so please do memorize that. Alright, that's it for today, and in our next video, we will be looking at the process of converting the G3P molecule into triose phosphate, which is also known as TP, and then from then on, we're going to look how TP is either used to regenerate RuBP or just kind of leaves the cycle. We're going to look at the process in our next video.